another very exciting area of developing applications in cancer therapies. And as you may know, here at UF, we have an extremely strong and prominent immunotherapy group that is, among other things, developing highly efficient dendritic cell vaccines and looking at different ways to make them even more efficient and safe. So here, the idea is that dendritic cells are being primed and activated with tumor-specific antigens, specifically to enhance the T-cell immunity. So the dendritic cells that were isolated from a patient are pulsed with tumor antigens, and so which leads to their activation and maturation. And after that, these dendritic cells can be re-injected into the patient, and now these dendritic cells can prime and activate cytotoxic CD8 positive T cells that start patrolling the body to find and kill tumors with that express the specific pretty much any of the tumor antigens. So the advantage here is by pulsing the dendritic cells with tumor antigens, we do not need to know the specific tumor antigen, and we do not need to separate even the self-antigen from neoantigens, because the self-antigens are already going to be tolerated by the immune system as a self-antigen, and only the tumor antigens are going to induce and potentiate anti-tumor response. So this is really useful for this agnostic use when we don't really know what tumor antigens are really present. And so one of the examples of FDA-approved dendritic cell vaccines is the Sipuleucel-T, which is used for metastatic castration-resistant prostate cancer. And this is pretty much a cell therapy in its principle. These are peripheral blood mononuclear cells that are being isolated from a patient, and they're being activated with a recombinant fusion protein. So it's a prostatic acid phosphatase, which is expressed in a lot of prostate cancers, and it's also fused to GM-CSF, so granulocyte monocyte colony specific factor, which would promote maturation of the antigen-presenting T cells. And again, to reiterate that, so the monocytes are being, mononuclear cells are being isolated from the patient, and monocytes are being selected out of all peripheral blood mononuclear cells. They're being pulsed with a fusion protein and cultured and reinfused back into the patient. And so now these infused monocytes, because they see the antigen and the GM-CSF, they mature and differentiate into activated antigen-presenting cells, which are now able to present the antigen to cytotoxic CD8 positive T cells that are now become primed and activated for direct tumor lysis. So another idea in engineering pretty much tumor vaccines are the engineered oncolytic viruses. And so right now there's one example that's been FDA approved. However, again, it's a actively growing area of research with many new oncolytic viruses being developed and tested, including here at UF by some groups here. So this particular example is being used for recurrent unresectable melanoma and has to be injected specifically into the tumor, unlike other immunotherapies that can just be reintroduced into the circulation. And so the idea here is why these oncolytic viruses are specific to the tumor. So let's think about it this way, that normal cells evolved to see a lot of viral assaults, and therefore normal cells have antiviral mechanisms that evolved over time in place to resist the viral infection, and either usually shut down the expression of viral mRNAs and viral proteins. And the interferon-based pathways are pretty much the central example of the mechanism. So tumor cells, as kind of the bystander effect in the tumorigenesis, oftentimes lose these antiviral responses, pretty much because the tumor is so focused on unlimited growth that these antiviral responses become almost superfluous, and can be shut down to not to interfere with active tumor growth. And therefore, now that this antiviral protection is removed, the oncolytic viruses can easily infect the cell, and not only infect but also replicate and lead to cell lysis. However, the normal cells that still retain the antiviral mechanisms are immune to the viral lysis. And so here the TVAC is exactly an example of this. It's an engineered oncolytic virus. So it can infect a lot of different cell types. However, it can only replicate in tumor cells. And so it is also engineered to carry GM-CSF, which is a granulocyte monocyte colony stimulating factor. Again, as we know from previous examples, it's a very important cytokine to drive dendritic cell differentiation and maturation. And so as the virus starts replicating specifically in cancer cells and leads to cancer cell lysis, now it leads to the exposure of a large number of tumor-specific antigens and also of the engineered GM-CSF. And so now these tumor-specific antigens are being taken up by the dendritic cells that are additionally being activated by GM-CSF to educate the T cells and drive additional tumor lysis. And so the T cells become programmed to destroy cancer cells anywhere in the body, not only in the primary tumor where the T-VEC was injected, regardless of whether they're infected with a T-VEC or not. So now we're going further and further, and with every type of cancer immunotherapy, I think it becomes more and more exciting. And now we're talking the immune checkpoint inhibitors, which I'm sure you heard a lot of, especially because of the relatively recent Nobel Prize that was awarded to Tsuku Honjo and James Allison, the two researchers that described the two different mechanisms of immune checkpoint. And so let's, again, very superficially, let's go over these mechanisms. So first of all, we've already talked about dendritic cells presenting the tumor antigens or any antigens to the T cell in order to activate and prime T cells. And so that's where it happens, where the antigen is being presented by MHC's major histocompatibility complex class 1 to the T cell receptors. However, in order for the T cell to become activated, it needs an additional signal. It means an activating signal, which either comes in the form of B7 or B72. So they're also known as CD80 or CD86. And this B7 co-stimulatory signal needs to be bound by CD28 on the surface of the T cell. And that is a signal for T cell activation. However, soon after T cell activation, the T cells start expressing CTLA4, which is now an immune inhibitory molecule that can also be bound by B7. And once that starts happening, that provides an inhibitory signal to the T cell to avoid the overactivation of the immune response. And so since tumor antigens are oftentimes present in high amounts in the body, it leads to the overstimulation of the T cells that leads to the activation of immune inhibitory mechanisms. And so that's where the CTLA4 is the major player. And so if we can inhibit the interaction, the immune inhibitory checkpoint interaction between CTLA4 and B7 by, for example, using an antibody, that would prevent the inhibitory signal and leads to and allow the activation signal, activation co-stimulatory signal, and lead to T cell activation. And then also so that happens in the priming phase and usually happens in the lymph node. And so now if we're talking about the effector phase, that happens in the target tissue, in the peripheral tissue. So again, the T cell receptor would recognize the tumor antigen presented by MHC class 1 on the surface of tumor cell. However, again, to avoid overactivation of the immune response, the T cells express the PD-1 receptor that can be bound by PD-L1, so the programmed death ligand 1, that is very often overexpressed on the surface of tumor cells because the tumor cells under the immune pressures evolve to express the immune inhibitory signals. And so that inhibits the T cell cytotoxic function. And if we can break up this interaction between PD-1 on the T cell and PD-L1 on the tumor cell in the peripheral tissue, that may prevent this inhibition and lead to and allow the T cells to carry out their tumor-lytic function. And so just because it's an important point, I want to reiterate that the CTLA4 inhibition happens in the lymph node, and it's the interaction between the dendritic antigen-presenting cell and the T cell, while the PD-1, PD-L1 inhibitory interaction happens in the peripheral tissue between the T cell and the target cell. And so currently there are 3 different antibodies that are approved for as checkpoint inhibitors. It's the ipilimumab, which is the first immune checkpoint inhibitor ever approved by FDA in 2011 to be used for stage 4 melanoma. And there are 2 different PD-1 or anti-PD-1 or anti-PD-L1 antibodies, pembrolizumab or atezolizumab. Are currently FDA approved either for melanoma or for bladder cancer. But now when we're talking about which cancers, how do we know which cancers are going to respond to immune checkpoint inhibitors? Because I'm pretty sure that you've heard a lot about either primary resistance or acquired resistance. So we know that for the immune system to effectively identify and target the tumor needs to be characterized by tumor-specific neoantigens that are different from the self-antigens. And so here, if we look at the prevalence of mutations, pretty much the extent of increased mutagenesis in different tumors, we can see that different tumor types vary greatly, more than 100-fold between the tumor type with the least number of mutations, of new mutations per megabase in the genome, to the highest tumor type with the highest number of novel mutations per genome. And so if we now correlate this mutation load per megabase in different tumors to the objective response that has been seen in patients during clinical trials and also in during preclinical studies. So we can see that generally the larger the mutational load in the tumor, the higher the rate of response. And so there is a pretty strong correlation that the more mutagenic the tumor is, the higher the response to checkpoint inhibitors, because obviously highly mutagenic tumors are going to be predicted to have a higher number of neoantigens that can be recognized by the tumors, by the immune system. And so here, for example, mismatch repair deficient tumors and mismatch repair proficient tumors would be a really good example, where mismatch repair deficient tumors, which will very rapidly accumulate large number of either point mutations or also short repeat expansions, are highly responsive to checkpoint inhibitors. So pretty much the only outlier in this graph would be the Merkel cell carcinoma, which is here. It has a relatively low mutation load per megabase. However, it still has a pretty high response rate. And so Merkel cell carcinoma is a skin cancer. It's the neuroendocrine carcinoma, and it's predominantly driven by presence of the poliovirus. And so even though there's a relatively low mutation load in the host genome, the viral antigens are the ones that are driving these high response to checkpoint inhibitors. And so I was talking about the mismatch repair proficient and deficient tumor. So if we're looking at colorectal cancer, which is mismatch repair proficient, which means that it has a low number of novel mutations, it has a pretty dismal response to checkpoint inhibitors. So again, if we were just to cover the PD-1, PD-L1 inhibitors, the pembrolizumab, for example, it activates the body's own T cells that have the capacity to recognize and eliminate cancer cells. So the T cells express PD-1, which helps to negatively regulate the immune response. And so the PD-L1 is commonly expressed on the surface of cancer cells that evolve under the pressure of immune system elimination. And so a lot of them evolve to express the inhibitory signals such as PD-L1. And so this interaction would put a brake on the immune system. And so if we can block this PD-1, PD-L1 interaction, that reactivates the cells and allows them to carry out the tumor cell killing function. And so finally, let's look at the chimeric antigen receptor T cells, or CAR T cells, and some people also call them CARTs. And so why it's called a chimeric antigen receptor? It's because these are T cells, so these are patient-derived T cells that have been engineered to express one molecule, which is a fusion or a chimera between the target binding domain on the surface, but then it was also engineered to have all the necessary co-stimulatory domains that are usually provided by separate molecules in the T cells. And here they were just all fused together to have an all-in-one design, to have the target binding and recognition domain and all the activation domains to unleash the cytolytic activity and cytokine release in the T cells. And so cytotoxic T cells would need to know which cells. So in order to engineer these cytotoxic T cells, first of all, we need to know the target antigens that we're trying to target, because this part of the molecule, the specificity that drives the specificity, is going to be pre-engineered. And so therefore, in order to have very effective CAR T cells, we need to have a universal tumor antigen, which oftentimes presents a challenge because only certain tumor types may be characterized by universal antigens. So also, so far, CAR T cells have not been particularly effective in solid tumors. It's mostly because of the highly immunosuppressive microenvironment that is being present in those tumors. And so now multiple attempts are being made to also engineer the CAR T cells to, for example, express certain cytokines that would help combat this immunosuppressive microenvironment. However, so far, there hasn't been a lot of success in that area. And also, currently available CAR T cells are only effective against T cell malignancies. So we've already talked about that there's a challenge associated with solid tumors. However, even with liquid tumors, so far, just the B cell malignancies are the huge success, while myeloid malignancies are a lot more challenging. And the reason is that a human can live without B cells. Yes, there are going to be problems with immune response. However, B cells are not, we can still live without B cells. However, humans, we cannot live without myeloid cells. And so in this case, because we don't really have the means to engineer CAR T cells that would be specific to just the malignant myeloid cells, as opposed to all myeloid cells. In this case, it really needs a suicide switch so that the CAR T cells can be turned off after they've fulfilled their tumor killing function. And so far, there's been a lot of challenges with the safety of these approaches, and they have not made it to the clinic yet. And so CAR T cell, again, first of all, it's a cell therapy. It's an autologous cell therapy, which means that these are T cells derived from a patient. And it's also a gene therapy. This is actually the first ever FDA-approved gene therapy, because now, once the T cells, the patient T cells are being isolated, they're being modified, they're being sent to a special manufacturing facility where they're being, where the CAR, the chimeric antigen receptor, is being delivered through a retrovirus, after which these modified T cells are being selected and expanded. And once the manufacturing is done, which usually takes about 3 weeks, they're being shipped back to the hospital to be infused back into the patient. And so now these, so currently available T cells are anti-CD19 specific T cells. So now they can start circulating and killing all of the CD19 positive cells. So we have here 2 examples of CAR T cells. They're both anti-CD19 CARs. So they were both approved very recently in 2017. So for B-cell pediatric and also adult relapsed refractory B-cell malignancies. And again, as we discussed, some of the major side effects would be associated with depletion of B cells and also with overactivation of the immune system. And so here are the toxicities that are being associated with CAR T cell therapy, which are prevalent. So they happen in a large proportion of patients, usually more than half to some extent. Some of them might be dose limiting, and even some of them might be deadly. So the overactivation of the immune systems, the so-called cytokine release syndrome, would pretty much present as a massive bacterial and or viral infection with dangerously high fever, precipitous drop in blood pressure, and oftentimes even with organ toxicity and neurotoxicity. And currently, the severe CRS, cytokine release syndrome, is being managed by tocilizumab, which is an anti-IL-6 antibody, as interleukin-6 is believed to be the primary cytokine driving the cytokine release syndrome. However, there's a lot of other cytokines involved too, and in really severe cases that involve neurotoxicity and acute organ injury, the steroids would also be used in combination with tocilizumab. And so other on-target toxicities would involve B-cell aplasia, again, as we discussed, it is massive B-cell killing and may lead to immunocompromised state and also reactivation of some endogenous viruses or hepatitis, such as hepatitis B virus. And that is being managed by mostly antibiotic cover, and also in severe cases by immunoglobulin infusions, pretty much to replace the loss of antibody-producing B cells. And so that concludes our lecture. So let's just have a look, revisit again the tumor immunity cycle, where it starts with a release of tumor antigens, oftentimes as a result of tumor cell killing, oftentimes usually as a result of chemotherapy or radiation or other anti-cancer therapy that leads to the release of tumor antigens that are now being taken up, processed, and presented by the dendritic cells. And so here, this is the target of either dendritic cell vaccines and cytokines such as interferon alpha and GM-CSF and other immune modulators. So the next step would be the cross-presentation of antigens and activation and priming of T cells that happens in the lymph node, where the dendritic cells are activating the T cells. And again, this is the target for some other immunomodulators such as interleukin-2, and also, as we discussed, one of the checkpoint inhibitors, the anti-CTLA-4. And once the T cells are being primed and activated, and they start circulating and patrolling the body, extravasate in the target tissues, and track down, find, and lyse the tumor cells. And so here, the recognition of the cancer cells by T cells can be directly manipulated by chimeric antigen receptors, while the killing of cancer cells can be unleashed by anti-PD-1 or anti-PD-L1 immune checkpoint inhibitors. And so that's it for today. I hope you enjoyed this lecture. And again,